It's about media design that increased high mannose glycans 2.8-fold in CHO cell culture runs. It works in multiple cell lines. It doesn't require cell line reengineering or expensive glycosylation inhibitors. It's not a universal fix. I'll tell you exactly when it works and when it doesn't. Welcome to the Smart Biotech Scientist. I'm David Brühlmann, and some years ago, I came across a study that stopped me in my tracks. Cyclists who ate pistachios before a 75-kilometer time trial performed worse than those who didn't. Not marginally, 4.8% slower. That's statistically significant. Why would a healthy nut sabotage athletic performance? The culprit, raffinose, a trisaccharide in pistachios. During intense exercise, your gut becomes more permeable and raffinose from the colon leaked into the bloodstream. Once there, it correlated with oxidative stress and leukotoxic effects. Your body is literally fighting the fuel you just gave it. I thought, if raffinose can slip through a stress gut barrier and trigger metabolic effects in humans, what does it do to CHO cells under bioprocess stress? That question led to one of the most surprising findings of my PhD work. Before we dive into that story, let me give you the roadmap for today. First, I'll walk you through why glycosylation matters for biosimilars and what happens when it goes wrong. Then, I'll show you the biology of how raffinose changes glycan processing. After that, I'll take you through the experimental journey, what we tried, what failed, and what finally worked. And I'll close with the real-world impact this had on actual development programs. But let me tell you what this episode is really about. It's about media design that increased high mannose glycans 2.8-fold in CHO cell culture runs. It works in multiple cell lines. It doesn't require cell line reengineering or expensive glycosylation inhibitors. It's not a universal fix. I'll tell you exactly when it works and when it doesn't. But if you're 6 to 12 months from filing, this could save your timeline. Before we go further, a quick primer for anyone who is new to glycosylation or needs a refresher. Glycosylation is the process where sugar chains get attached to your protein as it's being made. Think of it like decorating a cake as it comes out of the oven. Except the decorations determine whether your drug works or gets cleared from the bloodstream in 10 minutes. This happens in two cellular compartments. First, the endoplasmic reticulum, or ER, adds a core oligosaccharide structure. It's a 14 sugar arranged in a specific tree-like pattern. That's the starting template. Then the protein moves to the Golgi apparatus, where the real action happens. Enzymes in the Golgi trim off some sugars and add others, sculpting that core structure into the final glycan pattern. For monoclonal antibodies, glycosylation affects a lot. Half-life in circulation, immune activation through Fc receptors, the protein stability, and most importantly for your biosimilar development program, whether regulators will approve your product. If you get it wrong, you're looking at 6 to 12 months of rework. You get it right, and you're on track for approval. Now back to raffinose and why it matters. Let me paint you a scenario that's all too common in biosimilar development. You've spent 18 months developing a biosimilar map. Your cell line is stable, titer is great, around 3 to 5 grams per liter, and you're hitting your productivity targets. The team is feeling good about it. Then the analytical team drops the bomb. Your high mannose levels are at 1.4%. and the reference product is at 6%. You're out of spec. Now, you might be thinking, why does it matter? It's just a small difference in sugar composition. Here is why it matters. High mannose glycans affect antibody-dependent cellular cytotoxicity, or ADCC. They affect receptor binding kinetics. They affect serum clearance rates, and most critically, they affect regulatory comparability. If your glycan profile doesn't match the reference product within an acceptable range, regulators will ask for additional clinical studies, or they will reject your filing outright. So what are your options? Well, option 1 is a temperature downshift to about 32 to 33 degrees. It can push the glycan profile toward high mannose, but the productivity trade-off is clone and process dependent. Option 2 is kifunensine. It's a glycosylase inhibitor that blocks mannose enzymes in the Golgi. It's effective. You get high mannose, no question. But it's expensive, hard to scale, and regulatory agencies scrutinize any process that uses enzyme inhibitors. You'll spend months justifying it in your CMC package. Option 3 is you re-engineer your cell line. You basically go back to the drawing board. You select a new clone with better glycosylation characteristics. This works, but it adds at least 12 to 18 months to your timeline. If you're racing to market against competitor biosimilars, that delay could cost you hundreds of millions in lost revenue. What you really need is a media-based solution. Something that scales, something that doesn't torpedo your productivity, and something that regulators understand because it's just a media component adjustment. That's where raffinose comes in. Let me explain the biology of what's happening when you add raffinose to your cell culture. Raffinose is a trisaccharide, 3 sugars linked together. It contains galactose, glucose, and fructose. CHO cells can't metabolize it efficiently, so it accumulates in the culture medium and in the cell. It turns out that raffinose competes with a specific enzyme in the Golgi. It's the glucan transferase. This enzyme is responsible for adding the first branch to the core glycan structure. Think of it as the fork in the road where a simple glycan becomes a complex, multi-antennary structure. When raffinose is present, it binds to the active site of glucan transferase. And what I must say, it doesn't shut the enzyme down completely, it just slows it down. What is the result? Your antibody ends up with mannose 5 species instead of the complex, fully elaborated glycans you'd normally see. And I must say, this is different from kifunensine. Kifunensine blocks mannose 1 and gives you predominantly mannose 8 and mannose 9 glycans. These are much earlier intermediates. Raffinose arrests glycan processing at a later step, giving you predominantly mannose 5 glycans. We confirmed this mechanism with transcriptomics. When we looked at gene expression in the cells treated with raffinose, we saw downregulation of galactosyltransferase and other late-stage Golgi enzymes. The cells were adapting to the metabolic stress by dialing back their glycan elaboration machinery. The key thing to understand is this: raffinose doesn't shut down glycosylation entirely, but it arrests it at a specific step. That's why you get mannose 5 enrichment and not a complete block. And that's also why it's tunable. Basically, you can dial the concentration up or down to hit your target glycan profile. Now, knowing the mechanism is one thing. Making it work in practice, that's where things got interesting. Let me take you through what we actually tried in the lab, because the path to breakthrough was not straightforward. The first attempt we were taking is just using standard media supplements. We started with the usual suspects. Manganese chloride, known to affect galactosyl transferase activity, galactose and glucagon at various concentrations, trying to push or pull the glycan pathway. We tested these in 96 depot plates across a range of concentrations. The result, well, minor glycan shifts. Nothing reproducible, at least for high mannose species. We also saw batch-to-batch variability, and there was no clear dose-response relationship. We were tweaking the system, but we weren't controlling it. Our second attempt focused on osmolality. We knew that osmotic stress affects Golgi function. So we tried increasing osmolality by adding raffinose at high concentrations, 50, 75, even 100 millimolar. The idea was to stress the Golgi and slow down glycan processing. The result, well, growth crashed. At 50 millimolar raffinose, the cell viability dropped below 70% by day 5 already. Then, as we were preparing for attempt 3, we had a realization. Because the raffinose concentration and the osmotic stress were two different things, and actually high raffinose meant a high osmolality in our medium, and we couldn't tell which factor was causing the growth inhibition. Was it the osmolality or the raffinose itself? So we made a constant osmolality pivot. This was the key experiment. We adjusted sodium chloride in the medium to keep the osmolality constant at 350 milliosmoles per kilogram, regardless of the raffinose concentration. So when we added 30 millimolar raffinose, we subtracted enough NaCl to maintain constant osmolality. What was the result? The growth recovered. The viability stayed above 90% through day 14, and we now could test raffinose concentrations up to 100 millimolar without killing the cells. Once we isolated raffinose effect from osmotic stress, the data became crystal clear. We had a dose-response curve. We had reproducibility, and we had a scale-up potential. It turns out that the breakthrough wasn't in finding a magic ingredient, it was in controlling the variables so we could see what raffinose was actually doing. Here is what worked. 50 millimolar raffinose increased high mannose 2.8-fold compared to the control. The effect was robust. It worked in 2 different cell lines, it worked in 2 different basal media, and it worked at every scale we tested in 96 depot plates, in shake tubes, and in benchtop bioreactors. We observed to our surprise that the glycan profile was predominantly mannose 5, not mannose 8 or mannose 9 like you'd see with kifunensine. This told us that we were hitting a different step in the Golgi processing pathway. Now, I was fortunate to apply this approach in actual development projects. I can share the specifics, those programs are confidential, but I can tell you this. When you're starting a glycan mismatch 8 months before IND filing, having a validated media lever in your back pocket is the difference between making your timeline and explaining to leadership why you need another year. One more thing about this approach that's often overlooked. It's regulatory friendly because raffinose is a simple trisaccharide, so it's available as a GMP grade material for multiple suppliers. It's not an enzyme inhibitor. It's not a genetic modification. It's a media optimization, something process development teams do all the time. If you're pre-IND, this is straightforward. Or if you are even pre-BLA while you haven't logged in your process yet, you're optimizing your process and media composition is part of that. Document it, include it in your development report, done. A long story short, we proved raffinose works. But here is the hard part. How do you actually implement this in your process without spending 6 months on design of experiment studies? In part 2, I'll give you the exact decision tree. when to use raffinose, when it won't work, and the 3 experiments you need to de-risk it before committing to your manufacturing campaign. If you want to dig into the full methods and data, the paper was published in the Journal of Biotechnology. You find the link in the show notes. Thank you for joining me in exploring the biology behind raffinose and the glycan control in CHO cell culture. If you found this episode valuable, I'd love your feedback. Share it by leaving a review on the podcast platform you found us on. It helps other scientists discover these insights and lets me know what's resonating with you. Thank you so much for your feedback. Thank you for joining us today, and I'll see you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.